Morning and good evening to Samrat. So it gives me great pleasure to introduce Professor Samrat Mukhopadhyay, who is going to give this morning's lecture. Samrat is currently a full professor at IISER Mohali. He did his PhD at the Indian Institute of Science in Bangalore and his postdoctoral work at the Scripps Research Institute. He's won a number of awards, and his most recent recognition has been his nomination as editor to Biophysical Journal. Samrat is a world expert in the field of intrinsically disordered proteins, and I think we can all say that we're very excited, Samrat, to hear your talk on an emerging area of research where you've really made a significant impact already, which is liquid-liquid phase separation. And Samrat, please take it away.
Thanks, Joan, for the kind introduction. And it's a great honor for me to talk about our current research in this wonderful ZOOMinar series. And I thank Joan, Rams, and Magda, and also Bikash for organizing this incredible seminar series during these topsy-turvy times. And thank you all. So let me first say that I have created some space on the top right corner of my slides for you to place the talking head so that it doesn't block the text or images on the slide. Okay. Well, I work at the Indian Institute of Science, Education and Research, Mohali, abbreviated as IISER Mohali, which is located in the suburbs of the city beautiful called Chandigarh, which is 150 miles north of Delhi at the foothills of the Himalayas. And it's a privilege to work at a place with a breathtaking view of the lower Himalayas. So I would like to first acknowledge, my incredible group. So I'm very fortunate to have very talented, highly dedicated students and postdocs. Some of them have already departed the lab, doing postdocs, PhD in the United States and Europe. My first student, Neha Jain, who worked with Matt Chapman for her postdoc at the University of Michigan. She is now back in India and as a faculty member at the Indian Institute of Technology in Jodhpur in Rajasthan. And of course, today I'm going to talk about something that we have been doing little over 2 years now. And this work was primarily carried out by Priyanka Dogra, who finished her PhD, now a postdoc with Richard Krivaki at St. Jude, and Anupa, a postdoc in the lab, and also Shiny, who was an undergrad in the lab, now a graduate student at UC Santa Barbara, and also Ashish, who was a PhD student in the lab. There are other students who are also contributing to our new research on liquid-liquid phase separation. And of course, we also have a significant effort in amyloids and IDP dynamics. I'm also thankful to IISER Mohali and other funding agencies for generously supporting our research over the years. And I'd like to thank Liz Rhodes, Jennifer Lee, and Jeff Kelly for their kind gifts of plasmids. And I'm also thankful to my wonderful collaborators and colleagues. So the overarching goal of our lab has been to understand the intriguing conformational excursion of intrinsically disordered proteins, particularly driven by the chain and chain solvent interactions, giving rise to this exquisite nanostructures, amyloids, and we ask questions how these diverse nanostructures are associated with debilitating neurodegenerative diseases like Alzheimer's, Parkinson's, prion diseases, and so forth, and how these oligomers, pernicious species are involved in the as the causative agents of the disease. And we are also quite intrigued by how these intrinsically disordered proteins can assemble into a wide range of nanostructures. And as I said, only in the last couple of years, we are also been immensely interested in phase separation of some of these intrinsically disordered proteins, and which is what I'm going to talk to you about today. And so we are incredibly familiar with the sequence structure function paradigm. And of course, most scientists make an implicit assumption that sequence drives the autonomous folding of a polypeptide chain into a form which is capable of performing biological functions. And of course, this particular paradigm has been challenged in the last couple of decades due to an emerging work by a large number of scientists. And now we know over 1/3 of eukaryotic protein contains intrinsically disordered proteins or intrinsically disordered regions. They do not follow this paradigm. They remain as conformational ensemble rather than folding into a well-defined three-dimensional structure. And they can, the conformational plasticity can allow them to adopt multiple structures and therefore allowing them to perform multiple functions. Therefore, it can expand the functional repertoire of proteins. And what is the most striking characteristics of these intrinsically disordered proteins? And of course, I have redrawn this plot called the famous Eversky plot. And here I've plotted schematically mean net charge and mean hydrophobicity. And if you have lower hydrophobicity and high net charge, of course, proteins, the polypeptide chains do not fold up into a unique structure. And you have the dashed line over here represents the boundary between the disorder and order. So my lab over the past one decade has made has been involved in looking at the dynamic signatures of intrinsically disordered proteins and how they convert into highly structured, highly ordered amyloid fibrils and amyloid oligomers. But today I'm going to tell you about phase separation, where polypeptide chains are essentially remain disordered and dynamic within this very unique assembly of IDPs. So a little bit of physical chemistry of intrinsically disordered proteins. So they, as I said, they're characterized by rapid fluctuation, interconversion, and structural ensemble. And the conformational preference is encoded in the sequence composition. And of course, if you have more charged residues, polar residues, and and of course, it is quantified by radius of gyration, and that represents the tug of war between chain-chain and chain-solvent interaction. On the left, you have predominant chain-chain interactions, allowing the polypeptide chain to form compact globules. On the other extreme, when the chain is embraced by solvent molecules or water molecules, you have an expanded coil. And when chain-chain and chain-solvent interactions are perfectly counterbalanced, what you get is an archetypal Flory-random coil, where scaling exponent is half. So this is the picture under infinitesimally dilute condition. And as you crank up the protein concentration and you get past a certain threshold concentration, then of course this chain-chain interactions can promote phase transition. So a growing body of current research has revealed that many cellular functions can be achieved by so-called non-canonical organelles termed as membraneless organelles, and they are formed via liquid-liquid phase separation of intrinsically disordered proteins or regions in the presence or absence of nucleic acids and other partners to form this liquid-like dynamic mesoscopic supramolecular assemblies, often non-stoichiometric supramolecular assemblies, and they are prevalent in many parts of the cells, including nucleus and cytoplasm. And they perform a wide range of functions, at least putative functions, and of course, so basically cells have a way to spatio-temporally control the condensation, carry out cellular biochemistry, and control a myriad of critical cellular functions. And as a physical chemist, we'd like to know this inner workings of these liquid droplets. We'd like to ask the question, how the conformational characteristics of this flexible, intrinsically disordered proteins can promote this biomolecular condensation into this liquid-like functional states. And of course, these liquid-like states can undergo further liquid to solid phase transition that is responsible for many neurodegenerative diseases. So, why IDPs are excellent candidates for phase separation? And of course, this is described by classic Flory-Huggins theory, where the enthalpic term, this is the enthalpic term comprising the Flory-Chi parameter or the interaction parameter associated with the non-covalent interchain interactions that counteract with the entropy of mixing, giving rise to positive Helmholtz free energy of mixing. Under that condition, you get phase separation of polymers, in this case, intrinsically disordered proteins. And of course, I've shown here a typical phase diagram as a function of volume fraction and temperature. And IDPs are biological associative polymers comprising an architecture of stickers and spacers, as described by Rohit Pappu and co-workers. So essentially, the sticker interactions are essentially the electrostatic, hydrophobic, hydrogen bonding, cation pi, and dipole-dipole interactions. Whereas the spacers interspersed by stickers are essentially, provide the dynamism to these condensates, and they allow the huddling together of the stickers and involve the making and the breaking of interactions, which is what allow the internal architecture to remain as liquid-like dynamic architecture. And of course, low complexity domains comprising repetitive sequence offer conformational heterogeneity and flexibility that allows the breaking and making of weak transient interactions. We have been quite interested in an IDP, neuronal IDP called tau, and we are interested in the K18 fragment of tau that has 4 oligopeptide repeat domains. And if you look at the NMR structure of PRE NMR structure of tau K18, you notice I've actually superimposed a large number of structures obtained by Martin Blackledge and co-workers, and you notice that there is a propensity to undergo collapse. Of course, it is not a collapsed protein like many other collapsed IDPs, but there is a propensity to undergo collapse. And we set out to ask the question how the conformational preference is important in the phase separation. And of course, Tony Hyman's lab and Marcus Wegstetter labs have shown the importance of liquid-liquid phase separation of tau in the context of microtubule nucleation. And of course, the conjecture is, when liquid-like tau condensates undergo solid-like transition, then they become pathological to neurons. So we have been quite interested in this system, particularly taking tau K18, and we followed the condition that was worked out by Marcus Wegstetter's lab. And indeed, we are able to form liquid droplets, and they retain all the key characteristics of liquid droplets. They are spherical, they undergo fusion, and if they undergo rapid FRAP recovery. And of course, after establishing that tau K18 forms liquid-like droplets, we set out to ask 3 distinct questions. Is there a shift in the conformational distribution during phase separation? Does the droplet interior recruit water? Is there an alteration in conformational dynamics of the polypeptide chain undergoing phase separation? In order to address the first question, we took advantage of the fact that tau K18 contains 2 cysteine residues, and we label them using pyrene maleimide, and they're separated only 31 residues apart. So there's a very short distance for doing FRET measurements, because regardless of whether the chain is collapsed or expanded, the FRET will be very high because of this short distance. On the contrary, excitostate dimer, as you see by excimer formation, long wavelength excimer formation, which occurs at a much shorter distance, typically less than 10 angstrom. Therefore, we postulated that we'd be able to detect any collapse or expansion during phase separation by looking at pyrene excimer fluorescence. By monitoring autonomous pyrene fluorescence from pyrene monomer and pyrene excimer, which can, when 2 pyrene moieties are less than 10 angstrom, you can observe a long wavelength excimer band. And we set out to perform phase separation experiments and monitored fluorescence spectrum. And you can see pyrene. Of course, I must say that we dope this system only by 1% of dual labeled pyrene tau K18. So it's basically you are looking at intramolecular collapse or expansion. So this is basically the shorter wavelength where you see vibrational vibronic bands. This is essentially the autonomous fluorescence emission of pyrene, and the longer wavelength corresponds to the excimer emission. And as you can see, as a function of phase separation, we saw a dramatic drop in the excimer fluorescence and a rise in the monomer fluorescence, which suggests that during phase separation, two pyrene moieties are moving away from each other, indicating a chain expansion. And of course, tau K chain has lower critical solution transition behavior. Therefore, you don't see any droplets below 15 degrees Celsius. And when we perform experiments below 15 degrees where there is no phase separation, we don't see significant change in the excimer monomer ratio. So these experiments suggested that collapsed monomers undergo a chain expansion, forming this liquid droplets. And of course, it makes perfect sense in terms of polymer physics principles. Interior of the droplets, of course, is a much better solvent than water because it's a polymer-rich, dense phase, and it is more like a theta solvent rather than a poor solvent. And of course, this expansion should allow the stickers to sort of get exposed and sort of talk to other sticker residues and form this liquid-like droplets. Therefore, we propose that there is a conformational expansion, so the solvent property in the droplet phase is likely to be theta solvent. Now the question is this a dry droplet or you have significant permeability of, water recruitment into the droplet? In other words, it's a permeable droplet. So of course, this was addressed using a standard fluorescence quenching experiment. So we label a tau K chain, the cysteine, using fluorescent 5-maleimide, and we use potassium iodide quencher and recorded fluorescence lifetime in the presence and in the absence and in the presence of increasing amounts of potassium iodide. And what we get is Stern-Volmer quenching, Stern-Volmer constant, and we can estimate the bimolecular quenching rate constant. And from these slopes, it is evident that in the monomeric state, which is the cyan color, you have much less quenching compared to in the droplet phase, which is the red trace, okay? And you can see the rate constant in the droplet is also much higher than the monomer, and this is free fluorescein. So this tells you that, of course, chain expands, therefore these residues have much better accessibility to water, and that also tells you that the droplet interior has a lot of water. In other words, it's a permeable droplet. And of course, potassium iodide being a water-soluble quencher, it can only quench fluorescence when water goes into this polypeptide chain. So these are there is considerable water recruitment. Now the next question is, what's the fate of the chain dynamics within the droplets? Are the chain dynamics dampened in the liquid droplets? In order to address this particular issue, we performed fluorescence anisotropy experiments, and of course, our lab has been working on developing and adapting fluorescence anisotropy measurements for a wide variety of IDP assemblies. So I have shown here a confocal microscopy image of the mixed phase where there is no droplet. And of course, as phase separation proceeds, you can see larger and larger droplets. And even after a long time, these droplets retain the ability to undergo fusion. So this is also used about 0.1% of fluorescein labeled tau KT. Now we set out to ask the question, what happens to fluorescence anisotropy, which is related to the rotational tumbling of the fluorophore attached to a polypeptide chain. And of course, this tells you how fast a protein is undergoing rotational tumbling. So, of course, we expected that as the phase separation occurs, you'll see slower and slower tumbling due to dampening in the dynamics. And when we measured steady-state fluorescence anisotropy, in sharp contrast to canonical expectation, we see a sharp drop in fluorescence anisotropy, which means that there is more rotational mobility within the droplets rather than in the monomers. Okay, it was sort of counterintuitive at that time when we saw the data. And of course, we did all the turbidity corrections, and those things are all taken care of. So these experiments suggested there is a possibility of having a much more mobile polypeptide chain within the liquid droplets. So how do we establish this? Of course, we perform more sophisticated time-resolved fluorescence anisotropy measurements. So these experiments are performed using picosecond laser, a train of picosecond laser pulses. And this time resolution allows you to separate different dynamic modes, like the fast local dynamics and the for the fluorophore, the local dynamics of the fluorescent dye, and the global tumbling of the entire biomolecule or the polypeptide chain. And you can separate this on the time scale from picoseconds to nanoseconds. And we, of course, this can be described by a sum of exponentials that sort of discerns different dynamic mode. And in the last, over the last 5 years or so, we have been quite interested in looking at time-resolved anisotropy to look at fluorescence depolarization of intrinsically disordered proteins. And we observed that when you look at intrinsically disordered proteins, because it's not a packed, cell-packaged, protein like a folded protein. So rotational dynamics is primarily driven by the dihedral phi-psi dihedral rotational dynamics of the polypeptide chain. And we have done some extensive work in this area, and we have summarized this concept and methodology in Methods in Enzymology a couple of years ago. So essentially, what it tells you is if you have a compact disordered protein, in other words, collapsed disordered protein, and if you attach a fluorophore and look at the fluorescence depolarization, you can observe the global tumbling of the collapsed globule, and that will also be consistent with the hydrodynamic radius, because that can be related to the hydrodynamic radius via Stokes-Einstein's law. Whereas for expanded polypeptide chain, now you have intrinsic dihedral dynamics going to depolarize the fluorescence anisotropy much faster than the tumbling of the entire protein, okay, because it's a swollen polypeptide chain. And of course, you see a characteristic time scale from fluorescence, depolarization measurements using time-resolved anisotropy. And there's a characteristic time scale of about 1.3, 1.4 nanoseconds, which we have extensively characterized. And we think it is basically the conformational excursion of proteins on the phi-psi Ramachandran dihedral space. Now, for the phase-separated droplets, what we see is quite interesting. So, for the monomeric tau protein, we see a long rotational correlation time that is consistent with its hydrodynamic radius with a collapsed or at least partially collapsed polypeptide chain. And as the tau undergoes phase separation, liquid-liquid phase separation, and you can see there is a much faster depolarization kinetics leading, so that basically there is a conformational transition or dynamical transition due to conformational transition from more collapsed polypeptide chain to more expanded polypeptide chain. So, which is, of course, these are all quantification of rotational correlation time and amplitudes and so on. So essentially, what we infer from these time resolved measurements is that in the monomeric state, you have a collapsed disordered globule which undergoes rotational tumbling, which depolarizes the fluorescence anisotropy, whereas in the droplet phase separated state, you have expanded polypeptide chain undergoing fast internal dihedral dynamics, which is a characteristic of IDP in a theta solvent or good, much better solvent. So this slide summarizes our observation, of molecular shape shifting during liquid-liquid phase separation. And as I've already mentioned, pyrene excimer fluorescence tells us the polypeptide chain is gets expanded as being recruited into the liquid droplets. And there is a lot of water recruitment in the droplets, so these droplets are permeable. Of course, this makes perfect sense because for functional droplets, you need permeability for carrying out reactions and so on within the phase-separated condensates. And we've also been able to show that there is conformational fluctuations, which drives this, sticker making and breaking of sticker interactions. And of course, there has been a suggestion that these fluctuations can also add an additional three-body and virial coefficient terms into the Flory-Huggins classical term, which tells you that these fluctuations can, in principle, provide more, favorable, driving force for phase separation. So I'd like to change the gear a little bit. So we have been quite interested in another intrinsically disordered protein for several years now. This is essentially the repeat domain of a functional amyloid protein called PMAL17. So this protein is very interesting. It forms functional amyloid in melanosomes at low pH. Of course, melanosome is a lysosome-related organelle, so pH is around 5, 4.7 to 5 or so. And it forms amyloid fibrils, which are used for templating the synthesis of melanin, which gives rise to skin color, eye color. And under cytosolic pH, these amyloids get dissolved into disordered monomers. So it's a reversible amyloid. There was fascinating discussion in the last talk by David Eisenberg about the functional and the difference between functional and pathological amyloids. So these are perfectly reversible amyloids. And we form these amyloids from this repeat domain, which is intrinsically disordered. And of course, this is a prediction, Ponder prediction, that the repeat domain is intrinsically disordered. We are also looking at other domains, but repeat domain is quite important for amyloid formation. So if you look at the sequence pattern, polypeptide, the amino acid sequence pattern, you'll notice a constellation of negative charge in the form of glutamates, okay, in the repeat domain. There are 10 repeats. and glutamates are there pretty much in all repeats except this one. And of course, this protonation of glutamate controls fibril formation, okay, and deprotonation, of course, dissolves because of significant electrostatic repulsion. And over the years, Priyanka has shown that this repeat domain forms a wide range of nanostructures, from dendritic structures to archetypal amyloid fibrils in the pH gradient from pH 3 to pH 7. Of course, in pH 7 you don't see any aggregation; they're all in monomeric form or non-amyloid form. And of course, this is one picture Priyanka is very fond of, that we can make melanin in the test tube by using these amyloid templates. Of course, by using the enzymes and the precursors, DOPA, you can make this beautiful melanin deposits in the test tube, okay, using these amyloid fibrils. So now one of the questions that we're interested in, that what happens to this repeat domain at pH 7, cytosolic pH. So Priyanka made a striking observation that upon incubation at pH 7, 7.4, 7.2, 7.5, upon prolonged incubation, she saw these beautiful liquid droplets. And of course, we much later, we went back to Monica Fuchs-Reiter's brilliant program that utilizes fuzzy protein fuzziness to predict quite a few things, including the probability of phase separation. And this is the threshold beyond which the probability of phase separation is very high. And this is now available in Monica's website, and anybody is free to use her software, which was published in JMB and in a couple of other journals. And so you can see the repeat domain of PMEL17. This is the repeat domain. LLPS probability, or liquid-liquid phase separation probability, is close to 1.92, which is well above this threshold line. In fact, this was very exciting for us. And then Priyanka did all these experiments to show that they form spherical droplets with fluorescently labeled repeat domain RPT. They undergo fusion, dripping, surface wetting. All these are key characteristics of liquid droplets. And of course, the movie helps. So here you will see a fusion. There is a fusion here between two liquid droplets, and so they are really dynamic and of course they undergo very fast FRAP recovery. So here is a FRAP trace, and you can see that these droplets, if you FRAP them, and they recover very fast. And of course, we did some of these experiments that we did for tau previously, like solvation experiments by doing quenching experiments, and then steady state and time dissolved anisotropy. Here as well, you can see that in the monomeric dispersed phase, the anisotropy decay is much slower compared to the droplet phase, which tells you that the polypeptide chain undergoes extensive dihedral fluctuations, and of course their expanded polypeptide chain. So we did all these experiments that we did for tau. And then we were interested in the molecular drivers of repeat domain phase separation. And as a function of salt, when we studied the phase separation, and we saw that under physiological salt concentration, around 150 millimolar sodium chloride, we get beautiful droplet formation. And as you increase the salt concentration, phase separation ability is diminished substantially, and you don't see any droplets beyond a certain high salt concentration. That tells you that electrostatic interaction is important. In this case, of course, at high salt concentration, there is electrostatic screening, therefore phase separation is inhibited at high ionic strength. So electrostatic interaction is important. And of course, there are glutamates, there are some other positively charged residues and so on, which can participate in electrostatic interactions. Next, we wanted to look at, this is sort of a serendipitous discovery. So we are interested in looking at tryptophan fluorescence. And of course, it has tryptophan, single tryptophan, great system to study, environmental, changes using tryptophan fluorescence. So when we study tryptophan fluorescence, this is the 0 hour, which is the monomeric dispersed, and this is demixed phase separated state. And we begin to see a shoulder. And this is definitely not tryptophan fluorescence, okay? And this is way away from tryptophan fluorescence peak, which is around 355 or so. And then when we sat at this wavelength and recorded the excitation spectrum, we clearly could identify a different excitation band. This is for the monomeric, you don't see that excitation band. For the phase separated demixed state, you see another excitation band in addition to tryptophan excitation band. So it's a novel, fluorescence, fluorescent chromophore that is now formed upon liquid-liquid phase separation, okay? Which has an absorption or excitation max around 320 nanometer, and fluorescence somewhere around 400, 420 nanometer. And of course, such fluorescence, of course, this is blue fluorescence, because you see blue visible blue fluorescence around 400 to 420 nanometers. This arises due to charge transfer via hydrogen bonded network of polypeptide chain. Now, if you sit at this excitation wavelength, not looking at tryptophan at all, you can now purely look at the charge transfer band without any shoulder from tryptophan, because this is what you're now exciting. And you can see, as a function of salt concentration, there is the charge transfer fluorescence gets diminished. And this is essentially a charge hopping via or the delocalized, sort of carbonyl NH peptide bond. So that gives rise to intermolecular charge transfer band, which has been shown previously by other labs. So for other protein aggregates, amorphous aggregates, and so on. And you can see this is a centrifuge tube. And as you increase the salt concentration, you can see there is this blue fluorescence. This is illuminated using handheld UV lamp. And you can see there is no blue fluorescence at high salt concentration where there is no phase separation. And of course, hydrophobic interaction is also important, because there are this, hydrophobic stickers, and we were also able to establish that hydrophobic interaction is important. Now we have electrostatic interactions, hydrophobic interactions, charge transfer interactions. Now the proposal is, if you change the temperature, you'll see a very interesting pattern, because electrostatic interaction is diminished at higher temperature, whereas hydrophobic interaction is much more pronounced at higher temperature. And at lower temperature is just the opposite. And when we do this temperature dependent studies, we see a very interesting thermoresponsive behavior, which is dual UCST, LCST behavior, giving rise to a loop-like behavior. Beyond a certain temperature, there is no phase separation. Below a certain temperature, there is no phase separation, indicating that both hydrophobic and electrostatic, like charge transfer and electrostatic interactions, are important. Therefore, we were able to show that at cytosolic pH, at pH 7, you have this droplets, and this is recorded by using DAPI channel in confocal microscopy. So you can see blue droplets, spontaneous blue fluorescence without any fluorescent tag. And at cytosolic pH, they form liquid droplets. At melanosomal pH, here they form typical amyloid fibrils. And we were also able to sort of distinguish amyloid formation that is characterized by high anisotropy, high ordering, low turbidity, because these are nanoscale aggregates, and high beta sheet content. Whereas liquid droplets are characterized by low anisotropy, high turbidity, and large amount of random wall structure because they are disordered, expanded, rapidly fluctuating. And the charge transfer through the hydrogen bonded backbone is shown. This is a cuvette having the solution of phase fluorescence cuvette illuminated with UV lamp. And this is confocal image, and this is, of course, a schematic representation showing charge transfer or electron transfer via hydrogen bonded backbone network, peptide network. Now, if this undergo a liquid to solid transition, this liquid droplets undergo liquid to solid transition. If they undergo a liquid to solid transition, you're expected to see a much more enhanced blue fluorescence, because now the backbones will come closer and they will form a very persistent hydrogen bonded network. And this is what we see upon aging. They lose this spherical shape, they become irregular shape, which means they undergo a liquid to solid phase transition, forming aggregates. And you can see typical tryptophan to charge transfer FRET, because now the backbones are coming together. Therefore, there is energy transfer between tryptophan to the charge transfer band. There is substantial increase in the charge transfer band because of more pervasive charge transfer in the solid-like network as opposed to liquid-like network. Of course, these are not amyloid fibrils. These are not like the amyloid fibrils that we observe at low pH. And the dynamism is also lost, as you can see, by very inefficient FRAP recovery as a function of liquid to solid phase transition, because there is freezing of chain dynamics, and you also see an increase in fluorescence anisotropy. These are all sort of putting together. There is a freezing of chain dynamics. So liquid-liquid phase separation is characterized by lowering of fluorescence anisotropy. In other words, you have more fluctuations, and aggregation or liquid to solid transition is characterized by the increase of fluorescence anisotropy, where chains are sort of stuck to each other and they can't move around. So what is the significance of phase separation? So we have been able to show that you have low pH regime, we have amyloid formation, and cytosolic neutral pH regime, we have phase separation. And we think this is how these IDPs are stored in the when it is not used for amyloid formation and melanin biosynthesis. So it might be a storing mechanism for this particular protein. And of course, when the pH is changed, from cytosolic to melanosomal pH, it undergoes amyloid formation, forms the amyloid, the melanin templates the melanin synthesis, and again it can possibly go back to the storing at cytosolic pH. So to summarize our results, so we have been able to show that as key molecular shifting events like chain expansion, solvation, and fluctuations are important for liquid-liquid phase separation. There's extended conformers in theta solvent, because inside the droplet you have a better solvent, And this environment can allow transient intermolecular contacts through a multitude of weak interactions. These are termed as stickers, and they can form a weak network of physical networks, physical crosslinks, sorry, and then rapid large-scale fluctuations can control the relay of making and breaking of these interactions, keeping this liquid-like state. Now, these temporal fluctuations are important, and of course, we have been able to show intermolecular charge transfer modulates phase separation, and it, of course, can promote liquid to solid phase transition. And when you have transient side chain contact, sticker interaction, you have liquid phase, and when you have persistent backbone contacts in the form of highly organized hydrogen bonding, we are likely to get a transition from liquid to solid phase state. These critical molecular parameters might also be important for mesoscale material property. We are interested in measuring some of the soft condensed matter properties, viscoelastic properties, rheological properties. And these are important because liquid-like states are more functional, dynamic, permeable, whereas solid-like states are more, pathological, and they are not really functional. So, so on this slide, I'd like to just, I'm almost done. So I'd just like to show the other activities, current activities going on in the lab. We are also quite interested in mitochondrial heat shock proteins and alpha-synuclein interaction, because alpha-synuclein aggregation toxicity also involves mitochondrial dysfunctioning. So we are quite interested in mitochondrial HSP and synuclein interaction. We have been quite intrigued by the Hofmeister effects in amyloid formation. A paper is currently under review where we have been able to show a very intriguing Hofmeister effect in amyloid formation. We've also been involved in Carli amyloid formation, functional amyloid formation. And recently, we have done some energy migration via HOMOFRET to study, to sort of get a two-dimensional correlation map in amyloids, which can essentially be used for discerning different amyloid strains and so on. Recently, we have also been quite interested in the prion protein and amyloid beta interaction, which is proposed to be one of the toxic pathways in Alzheimer's disease. And of course, prion protein lipid interaction is also an important phenomena in prion toxicity. And the other exciting thing that we have been interested in looking at this ultra-fast fluorescence, time-dissolved fluorescence spectroscopy to characterize internal friction of intrinsically disordered proteins. And we are also quite intrigued by yeast prion strains by looking at the dynamic characterization of the yeast prion protein by using ultra-fast time-dissolved fluorescence characterizations. And we are also involved in quite a bit of Raman spectroscopy to understand different strains, amyloid polymorphs, and so on. We have got some really nice data on surface enhanced Raman spectroscopy to characterize amyloid oligomers, intrinsically disordered proteins, and so on. Those are ongoing work in my lab. I thank you for your kind attention.
Thank you very much, Samrat, for a beautiful talk. So now we're going to move to the question period. And Samrat, you'll see a chat box. And maybe we can start with a question from the chat box. And if you could read a question out loud.
Is this the chat box?
Yes.
You see in the Q&A folder.
Yes.
Sorry, I said, yeah.
Yes. Yes.
Oh, sorry, Q&A, sorry.
Yes. So, so from the Fabel structure, it became obvious that K18 is missing in 6A. Did your K18 construct contain this additional 6 amino acid? I think it doesn't have this additional 6 amino acid. So I'll have to go back and take a look. And we're using a typical K18 sequence. I'll have to just go back and take a look, and I can come back to you later. And the next question, yes, this is an important question. The hydrophobicity of pyrene is a concern, yes. So that's the reason we are using only a fraction, very little fraction, 1%, typically 1% dual label protein for phase separation, and 99% is unlabeled. And we have been able to show that all the other characterizations are fine, in the presence and absence of fluorescent labels. All the, critical saturation concentration, temperature dependence, all of these properties are similar in the for no label doping and with label protein. So we believe that there is not much perturbation in the system. So there were no. So I'm reading the question, Joan. Is that okay?
That's perfect. And so why don't we actually move to a live
it's fine.
question?
Thank you very much.
Oh, Christian is promoted. Great. So let's move to Vijay. Are you on? I think you've been promoted, Vijay.
Can you hear me?
Yes.
Yeah.
Okay, great. Hey, Samrat, good to hear you and good to see you also.
Yeah, hi Vijay, how's it going?
Good, going good. I know, as beautiful talk, beautiful work. And I have a sort of a curious question about tau. And you have beautifully shown this conformation dynamics of tau K18 into this liquid phase separation. So as you know, Marcus Zwexter has demonstrated that these droplets eventually sort of get gelated and moves into an amyloid fibril stage, right? My question to you is that, do you think that these liquid droplets are obligate intermediates of fibrils, number one? Then if you, I assume not, I'm not sure, but I think I would like to have your go at it. But if you assume so, and if you think so, now what have you seen this transition occurring in your dynamics, and what would be the fate of water in that?
Yes. So beautiful question. Yes. So under certain conditions, this can undergo amyloid transition. And of course, RNA is a very important player. So Songyi Han has done some beautiful work on this particular aspect of tau phase separation, and in our under our condition, the liquid droplets, even after long incubation, we were not able to see amyloid fibrils, typical amyloid fibrils. And of course, there are some increase in thioflavin T fluorescence, but they don't really form typical amyloid fibrils under this condition. So now we are also looking at RNA and a few other protein cross interactions. So yes, the water, I'm fascinated by water. And I think what might happen is when the chains are now coming together to sequester into much more intimate association, then of course there is entropic loss in water. In fact, Shruti, my former PhD student who is now at UCSF, who was at UC Santa Barbara, now at UCSF, has done some really beautiful work looking at femtosecond spectroscopy to look at water dynamics in polypeptide chain. And we have been trying to do this, femtosecond experiments in the liquid droplets. I think there'll be something really remarkable if you can do it, to look at water desolvation, water dynamics, and so on. Thank you.
Great. Thank you, Samrat. And I'll probably get back to you after the session is an informal session.
Sure.
Thank you. So our next question is from Charlie Brooks.
Sure. Hi, Charlie. How are you?
Actually, very nice talk.
Thank you.
So my question is that, recently I've been...
That often, can you still hear me?
Yes.
I think I just switched to a different microphone.
Okay, no problem.
Okay. Often it seems that there are multivariate
critical for formation of these membraneless sort of organelles. And in some cases, they come from, nucleic acid, RNA, et cetera. In the context of tau, for example, in a cellular environment, are there other components that are leading to multivalency, or is all of that these sort of stickers and other parts of the peptide sequence themselves that are giving rise to the ability to form, this organelle and hold it together with what, in my view, is by physical cross-linking due to interactions across the species?
Yes, so this is a great question. In this, of course, what we have done, we have not used any RNA or other protein partners, and of course, we can build the complexity of this liquid phase-separated droplets using, sort of simulating the intercellular environment. And when you have RNA, and of course, it is known that RNA participates in phase separation of tau. And there are these electrostatic interactions between tau and phosphate of RNA that is important. As you can imagine, this is heterotypic interaction is important. And in another system, not in tau, in another system, we have seen that RNA can also change the thermal responsiveness of the phase behavior. So you can change the temperature. In fact, this has been seen by others for FAST, for example. If you take only FAST, it undergoes phase separation at a certain temperature. The moment you have RNA under physiological conditions, they can form phase separated droplets. So and I think these interactions modulate this temperature dependent phase behavior quite a bit because you have hydrophobic tug of war between hydrophobic effect, electrostatic, hydrogen bonding interactions, and so on. So yes, so RNA is something that we are thinking of looking at, especially to look at dynamics, water, and so on.
Thank you.
Thanks, Joy.
Oh, sorry, our next question. Oh, so I should say that it's a little bit past 8. So we're going to close the overall session, but everyone is welcome to stay on if you have further questions for Samrat. And we have a question well, first, let's thank Samrat one more time formally.
Oh, thanks.
For his
Thanks,
lovely
Dawn,
talk.
others.
And for those of you who wish to say, please, Samrat is willing to stay on and answer questions. So let's move to Bin's question.
Hi, Ben. Hi, Ben. We follow each other on Twitter.
Hi, very interesting talk. I have 2 questions about
Sure,
your talk work. So you're using chemical modification approach to put fluorophore. K18 construct has 2 cysteine. How do you manage to specifically label, to differentiate between one labeled cysteine or both of cysteine are labeled? How do you get that? Because you want to get.
Sorry, there is a delay, so I'm sorry about that.
Yeah, about the cysteine labeling on your tau K18 construct, there are 2 cysteines. So and you in your construct seems like both tau are labeled, but it could be maybe possible maybe only one cysteine are labeled. How can you separate singly labeled cysteine versus dually labeled cysteine?
Yes. So yes. So for pyrene excimer studies, we of course use dual labeled, and of course we can check the labeling efficiency by by absorption spectrum, and we know that it is dual labeled. For other studies, we use sparse labeling condition to label only one cysteine. And we now have single cysteine mutants And those single cysteine mutants are incredibly useful for looking at, site-specific, regio-specific information about the phase separation, about the specific, regions. So we do have single cysteines over tau 18, and now we are also looking at full-length tau. So we engineer these things using, doing site-directed mutagenesis and so on. And we do, sort of a good sort of, I would say, chemistry, bioconjugate chemistry to sort of ensure that, once both cysteines are labeled when necessary, and one cysteine is labeled when for that particular type of experiment. It depends on the type of experiments. But now we have single cysteine mutants which can be used very easily for site-specific labeling.
Yeah, I agree. I think a singly labeled the cysteine mutant will be good control for your overall system. The second question I have is more general. So you have the this amyloid transition from a monomer to phase separation, maybe possibly go to fiber formation. Will you be able to to detect the signal transition, like from your fluorescence spectrum, from like a phase separation to the amyloid formation, or directly from monomer to fiber formation? Because one of the thinking is the fiber formation, so it may precipitate out from the solution, and whether your method can detect that change.
Yeah. So yes, that's a great question. So using fluorescence anisotropy, we have used for the past, about 10 years. We have developed this fluorescence anisotropy methodology to sort of look at this disorder to order transition, okay, for amyloid. So we know what is the signature of fluorescence anisotropy for amyloid fibrils. And as we have shown that when it undergoes phase separation, you have much lower fluorescence anisotropy because of fluctuations. And so if there is precipitation of amyloid fibrils, we cannot study by fluorescence or any other spectroscopy for that matter. So we tend to sort of use dispersed or fibrils that are not really overgrown or precipitated out of solutions. So these are still sort of dispersed amyloid fibrils in solution, and fluorescence anisotropy will be very high. And if you do time resolved anisotropy, there'll be no depolarization. We have some of these traces in our recent biophysical journal paper that was, we did homophoretic measurements. You can look it up, or I can send it to you. We are on Twitter, so it's easy to get connected. So I'll send you the, our biophysical journal paper, the latest biophysical journal paper. You'll see that, the depolarization is not complete because there is a residual anisotropy that is due to very slow rotational tumbling of amyloid fibrils.
Thank you.
Thanks, Ben.
Thank you. So we have a few questions, Samrat, on the
Can
chat.
I ask a question?
Actually, yes, go ahead.
Okay, Samrat, great talk. This is Rams here. I wanted to ask a question about, because electrostatic interactions seem to play a big role in this phase separation. What about free lipids or interaction with the lipid membrane? I know you showed for synuclein. What about for this IDP, other IDPs? And the other question I have is, this phase separation, does it depend on the concentration? Does it form a micelle-like aggregate before phase separation?
This is actually the second question I'll answer first, because that's a very important question, and I've been thinking about it for a while. So phase separation, in some sense, it's different from aggregation, because aggregation will bring them much closer. So now we have spectroscopic signatures to sort of characterize that. And if you do NMR, HSQC measurements, for example, and we want to do NMR, and if you, I'll probably write to you, there are some really beautiful HSQC measurements and other measurements that can be done. So So, so all the spectroscopic signatures tells you this association is not very persistent. It is rather transient and sort of like, you know, coming to making and breaking of the sticker interaction. Therefore, I don't really know whether it goes via this typical oligomerization like micellization and so on. And I think, of course, one can take a look at this early phase of phase separation, early kinetic phase of phase separation, which we have not looked at. And of course, there are two types of kinetics, which you saw, saw in the, spinodal transition and, binodal. There are two types of, there is one nucleation dependent, the other one is sort of like nucleation independent. And so this is a great question. In fact, I can, work on it for the next 5 years to answer this question, whether it undergoes micellization, but I don't think they undergo some transition to so-called oligomers. I think oligomers are already kind of much more well-packed than these droplets. Okay. But there has to be some, as physical chemists, we probably have to have more quantification, like internal viscosity, rheological properties, and so on. So I think some of this quantification might actually be quite useful.
What about the free lipids?
Yeah, the lipid question. Sorry about that. Yes, of course, lipid. I don't know whether lipids, because we are looking at membraneless organelles, and under this condition, there is no membrane biologically. But it is possible that in the presence of non-aggregated monomeric lipids, there might be some phase separation, because we can sort of neutralize the sort of electrostatic repulsions, which can promote hydrophobic association. They can commingle into sort of like heterotypic, assembly. But we have not really looked at it. But that's something which one can possibly take a look at a very low monomeric lipid concentration.
Thank you, Samrat.
Yeah, I think that's something which you should look at.
Thank you. So let's go to, and so I just have your username, VPIN.
Yeah. Hi, Samrat. Very nice talk.
Thank you.
I have two questions. I missed the point how you assign the motions occurring on the 1.2 nanoscale nanosecond time scale to phi-psi fluctuations. Is there a possibility of other motions occurring?
Yeah, so that's a great question.
Specific motion you assign.
Yeah, so we have, we had a paper in Biophysical Journal 2016, which describes the entire step-by-step sort of, and of course in methods in enzymology. So we have been able to show that this 1.3 nanoseconds, 1.2 to 1.4, this time scale is related to a collective dihedral dynamics, because dihedral dynamics is also going to contribute to the rotational mobility of the polypeptide chain. And by looking at, so if you look at an intrinsical, there is some intrinsically disordered polypeptide chain. There is something called, the Kuhn segment, okay, which comprises about, let's say, 7 residues, okay. So if you look at the Kuhn segment, let's say 14 residues, and if you look at the dihedral dynamics, of course, dihedral dynamics is much faster on the intrinsic, if you look at carbon-carbon single bond rotation, that's on the tens of picosecond time scale. But then this tens of picosecond time scale is not able to completely depolarize the fluorescence, because there is, steric clashes. So it can't rotate completely, right? So it has to really wait for the next residue, and it depends on the sort of like the mobility on this particular, statistical segment of this polypeptide chain. And it turns out that is, this 1.3 to 1.4 nanoseconds actually an intrinsic characteristic time scale. And this is also consistent with the reorientation timescale of from NMR. So yeah, we have actually done quite a bit of work on this, more than 5 years ago to characterize that.
I'm just wondering whether one can assign a segmental motion to it. So it doesn't necessarily have to be a fluctuation of the peptide planes, but an entire segment actually fluctuates very fast.
Segmental motion is essentially the phi-psi dihedral dynamics, right? Segmental mobility has to be, primarily for phi-psi dihedral dynamics because it has to involve backbone, right? So.
Okay. This second question that I want for the PMEL17, what determines the transition from weak side chain interactions into strong backbone interaction? You showed that at 96 hours approximately this happens, or 72 hours. So what is the driving force for that?
So we think there is this local nucleation that can happen within this phase separated state, because now they are sort of confined, into a very small space, and this local nucleation can, take place at a much longer time scale. And when this nucleation events probably start somewhere in the droplet, and then of course it sort of propagates into the other parts. The moment you zip a little segment into a much more closely associated hydrogen bonded structure, and of course that can propagate the transition, and of course then it can undergo liquid to solid phase transition. And of course biology is always trying to prevent that, because you don't want that transition, because liquid to solid transition is essentially a pathological transition for most cases, in most cases.
So as I understand, what you're trying to say is that basically you're trying to mimic sort of an amyloid formation, nucleation kind of an event. You're trying to map it onto this situation, but technically all your the liquid phase droplets are independent, and based on the data that you showed, I got the impression that each of them independently converts into these solid phases. So is there sort of a more concrete evidence to this, or this is sort of a general hypothesis?
This is sort of a general hypothesis, and using other molecular drivers, you can possibly prevent that. For example, there are beautiful papers by Max Planck Dresden group, Albert E. Hyman's group, that RNA can act as buffer. So basically, RNA can promote phase separation at some stoichiometry, at other, higher concentration. It can also, prevent liquid to solid transition. So we haven't really looked at that, and my guess is that RNA will definitely, sort of buffer phase transition, and it'll probably allow it to form more, liquid-like transition. So yeah, there are other, of course, when you're looking at a typical biomolecular condensates, there are other proteins, other RNA molecules which can play a pivotal role in maintaining the mesoscale material properties of these condensates.
Okay.
Thank you, Samrat. So we are getting past 8:15, but I think we can take, Samrat, if you're willing to take one more question, maybe.
Sure.
So I see that Vijay has his hand up again. Did you want to ask a second question?
Joan, Sahil is waiting, so can you?
Oh, Sahil, sorry. I'm sorry, Sahil. Please, Sahil, go ahead.
Yeah, hi, Samrat. Yeah,
hi.
I want to know I want to know how much of, what is the concentration of the tau K18 fragment you have used in your experiment, and what is the effect on this all your result when you increase the concentration of tau K18?
Yeah, so we typically tend to use tens of micromolar concentrations. And in some experiments, we have used 100 micromolar concentration for because they, for fluorescence experiments, we've used 100 micromolar. But yeah, typically I would say tens of micromolar. And under certain conditions, it can form, phase-separated droplets even at a lower concentration, a few micromolar concentrations.
So I have another question. So in phase-separated molecule, like in droplets, protein can achieve very high concentration compared to the normal cellular concentration.
You mean the interior concentration?
Yes, intrinsic. Yeah. So like for a function or for any protein, how for in vitro study, how can we decide a concentration if it's forming liquid droplets?
Yeah, right. So what the concentration that we operate at is basically the bulk concentration we are talking about, but the condensed phase concentration we have measured using, and this is an estimate, of course, this from confocal imaging, gels, we can estimate, and it is typically, hundreds of micromolar concentration. Okay, so it is much more concentrated, but not really highly concentrated like polymer melts or of that sort. So, because as we have shown that there is also a considerable water recruitment. So the dilute phase is called sort of like, you can call it as semi-dilute. This dilute phase is still maintained within this liquid drop. But in amyloids, for example, interior is pretty much dry, especially where you have, zippers and all that. And very little amyloid, very little water into amyloid structures. Therefore, there is a change in the concentration, the local concentration from liquid state to solid state.
All right, thank you. So let's maybe have the last question by Vijay.
Yeah.
Okay. Can you hear me again?
Yeah. Sure.
Okay. Hey, Samrat, I think I'm intrigued by the point that I raised, and I just want to come back to that. I think you mentioned that when you look at amyloidogenic proteins like tau, and when they phase separate, it makes sense to think of concentration gradient increasing inside the condensates, and then that will propel them to further aggregate, right? So that's what Marcus has shown, that it's beautifully on fibrils. Now, when you say that this in your observations, you've not seen that happen, I think the same transition we see with TDP-43 in our lab, right? So TDP-43 gets into this sort of stress granule type of situation with RNA. When you actually put that in, that they actually transition into fibrils pretty comfortably. But again, so some of the proteins which are not amyloidogenic, which probably makes a lot of sense to be in the liquid condensate form and then dissipate back into the same one single phase. But I'm curious to see why as you have not been able to identify or see a tau transitioning into the solid phase.
Yeah, so we have not looked at it for long enough. And we have not added other things which will allow it to form amyloid-like fibrils. So, and what happens is in some cases, you form this liquid droplets and there is a nucleation to form amyloid hydrogels, right? So, and it's not that entire, population of protein will undergo amyloid transition. There will be probably amorphous aggregates. There will be probably even liquid-like, phases. But in this phase-separated form, they can undergo this transition. But we have not. We're now looking at full-length tau, and tau K18, of course, can form solid-like aggregates. But whether they are typical amyloid fibrils, it's still, I think, we'll have to still look at it. Because by using most, standard techniques, we don't see it to be typical amyloid fibers. At least part of it can be amyloid, but
Yeah, understandable. Yeah. Thanks, Samrat. Great talk.
Thanks, Vijay.
Thank you. Thank you to
Thank you,
everyone.
Samrat. Yeah.
Thank you,
Thanks.
Samrat.
I think we're going to wrap up because we're nearly at 8.
Yeah.
Oh,
Thank you very
someone
much.
down.
See you later.
Yeah.
So thank you, Samrat.
It's done.
Thank you. Bye.
Thank you.